Okay, I think we are ready to start. So again, thank you very much for joining us today and welcome. In this webinar today, we are going to be talking about image integrity. So I'm going to introduce our speakers for the day. First, we have Jessica Offenberger, who is a publishing editor on the Sage Journal's editorial team. She works closely with editors and societies to help navigate the publishing process and develop key strategies. Next, we have Elaine Ellerton, who has been with Sage for 5 years, working in the open access STEM editorial department. She currently manages 2 journals, Neuroscience Insights and Science Progress. We also have today Adia Misra, who is the Research Integrity and Inclusion Manager at Sage. Next, we have Grant Maxey. After graduating with his DVM from the Western College of Veterinary Medicine in Saskatoon in 1969, Dr. Maxey earned a PhD in clinical pathology at the Ontario Veterinary College in 1973. He became board certified in anatomic pathology by the American College of Veterinary Pathologists. He was the director of the Animal Health Laboratory at the University of Guelph for 22 years. Grant was the editor-in-chief of the Canadian Veterinary Journal and has co-authored chapters in and edited the 5th and 6th editions of Jubb, Kennedy, and Palmer's Pathology of Domestic Animals. He is the current editor-in-chief of the Journal of Veterinary Diagnostic Investigation. Next, we also have Paco Yuzol, who is a professor of diagnostic pathology and branch chief of the San Bernardino Laboratory of the California Animal Health and Food Safety, UC Davis. He trained in Argentina, Sweden, and Australia. Dr. Yuzol has published around 340 articles in peer-reviewed journals. He is a diplomat of the American College of Veterinary Pathologists and a member of the American Association of Veterinary Laboratory Diagnosticians. He was the president and founding member of the Latin Comparative Pathology Group and president-elect of the same organization for 2 additional years. He was president of the Davis Thompson Foundation, and he's currently the chief executive officer of the same foundation. Dr. Yuzol is an associated and photo editor of the Journal of Veterinary Diagnostic Investigation and associate editor of Anaerobe Veterinary Research Communications and Microbiology Spectrum, and member of the editorial board of several other journals. And finally, there's me, Maria Tissot. I am marketing manager in the author marketing team at Sage, and my work is focused on helping early career researchers get published. You can send us your questions throughout the webinar using the Q&A box on your screen. We will have around 20 minutes at the end of the session to answer them. And if we don't have enough time to answer all questions, don't worry, we will follow up with you after the webinar. So now I'm going to pass it on to Jessica to start the presentation.
Thank you, Maria, and thank you everyone for joining another How to Get Published session here today. As Maria mentioned, we're going to focus on image integrity and what that means, how to edit your images ethically and or not edit them. So to get started, what we'll cover in today's session, we're going to cover image size, formatting, appropriate resolution, all of the image guidelines that you might want to follow, what type of image edits are acceptable and which ones are not, how to disclose those edits and adjustments that you might make, and how you should label your figures, your images, and what type of legends you should include. Just a few key reminders. The number one thing that we've been emphasizing, please make sure to read the journal guidelines to which you are submitting. Every journal has slightly different formatting requirements, and this also applies for images, but these are some general best practices that you can follow. So figures and images must be submitted as separate files and should be labeled accordingly. You do not want to embed them in your text document, your manuscript document. You'll want to submit your figures as TIFF files or JPEG files. They should not exceed 5 MB per file, and there's different compression techniques that you can use. So you can use the LZW compression to reduce that file size. If you do have layers in your images, do not flatten them before submitting to allow for editing if needed. So Dr. Rizal is a prime example of someone that might need to do some touching up on your images before it gets accepted and ultimately submitted to production. And then we'll go through some examples today, but if you're grouping those images into a composite figure, you'll separate each image with a thin white line. Don't worry, you'll see some great examples today of these best practices. Some tips on converting figures. Again, we're going through some of the high level overviews here. When you're looking at your image or your figure, it may not be in that TIFF or JPEG format immediately. So as you're going through your figures and potentially resizing things, cropping things, or making edits, you'll want to use Photoshop or another image editing tool to help with this. Some people do use PowerPoint to help put these images or figures together, for instance, like making composites where we talked about dividing with that thin white line. It is an option, so we always like to mention that, but most people will be familiar with Photoshop or another image editing tool. Something else to be aware of, for SAGE in particular, most journals will publish the figures online in color at no charge, and then it will be printed in black and white if there's any print versions of the journal going out. This is typically standard unless the authors opt to pay for those color images. Again, this will be clearly stated in the author guidelines if color figures are included in print or not. So be mindful of that as you're putting together your figure or your image, thinking about how that will appear in color versus black and white. And our team will go through some of those best practices today. Over to you, Paco, to go through best practices.
Thank you, Jessica, and good morning, everyone, or good afternoon, or whatever applies to your area. Some of the things I'm going to say apply to the journal we work for, which is the Journal of Veterinary Diagnostic Investigation from SAGE, and things may vary between journals, but usually they are pretty standard. So one is the resolution. We want images no less than 300 pixels per inch. This is for photographs, for if you take photos of gross specimens or histology or any specimen, try to make it no. I haven't seen any requirement for higher than this in any other journal, but I haven't seen anything below that. So usually 300. For line art, and Dr. Maxi will talk a little bit more about that, we require a little bit higher definition, 800 pixels per inch. And Jessica, if we can move on, please. Yes. So what happens when we at the journal receive images that are not big enough? More graphs or most line art, I can resize them for you. Let's say you send the line art, which is 300 or 400 dpi instead of the 800 that we require. It usually works, and I can enlarge it, and I'm going to show you a few examples. However, for photographs, it doesn't work because we lose resolution. Let's look at some examples. So this is a graph that was sent to us at 72 dpi, so well below the required resolution. And I digitally enlarged. In this case, just to 300 dpi. I could have done it to 800. And there's usually no loss of no loss of definition. This is probably, this picture, since we are at it, has several issues. The numbers are very little, they are difficult to read. But talking about resolution, I just wanted to exemplify that if it's too little and if it's line art, I usually can manipulate it myself. However, Jessica, over to you. See, this is a microscopic photograph of that thing that you have there is a blood vessel, and it's too little. When I enlarge it, see, I took it to 300 dpi and see how blurry it is. It loses definition. So that's why in this case, I have to go back to the author and say, please take a new photograph, higher resolution, and send it to me. So bottom line, line art, we can enlarge digitally, photos we can't. And Grant, over to you, I believe.
Thanks, Paco. Yes, and line art, again, similar to photographs or other images, people are trying to put their best foot forward. And you can do that. The line art that Paco just showed you was really busy. Try to not have it as busy. And the figures should stand alone from the text narrative. Sorry, I've got a train going by in the background. If you're using color, you should be aware that about 10% of men are either red, green, or blue, yellow colorblind. And putting a lot of color in graphs, line art, is not useful. It's not necessary. The clearest distinction, the best contrast, is black on white. And that's really what we prefer. So it comes in with a lot of colors, especially if you have things like purples and reds. A lot of men are not going to be able to distinguish those features. The incremental numbers on the axes should be minimal, should be 5 or 6, not in that one that Paco showed where you've got 15 or 20 along a line, really too busy. So you're trying to make some emphatic points here rather than incredible detail. We use sentence case for axis titles, and we show units of measurement. So again, it should be comprehensible at a glance to readers. If there is a composite figure, then we label in the lower left corner 1, 2, A, B, etc. If the colors will not appear in print, little lines with shapes. So squares and circles and open circles and so on, or pyramids, work really well. They're very clear to a reader. The abbreviation should be spelled out in the figure legend rather than on the figure itself. So we've got a probability figure there, which is okay. To give long formulas and definitions of IBD and TAC and so on makes the graph too busy. So simplicity is the key. You're trying to give maximum information with minimal input here to readers. Okay, thank you.
So I'm going to talk about modifying images, which is a pretty big part of this talk. Should you modify your images? And basically, you really shouldn't if you can avoid it. We do realize that sometimes it is necessary, or making some small changes here and there. So I'm going to talk about some of the things that are acceptable and some of the things that are just not ever acceptable. So as I mentioned here, it may be okay if you're just trying to simplify your image or make it clearer. Unacceptable modification is if you are actually removing any data or enhancing data and it no longer matches the original image that you have taken. And even if you think that you're just doing something to make it look better, it really is a breach of publication ethics. And I think it's really important to see that quote there. Digital images are data and they should be treated as such. So you really want to avoid modifying images if you can. Now I'm going to go over some examples of things that are okay and things that are not okay. So next slide. Okay, so some simple adjustments are okay, and there are some guidelines. As I mentioned, you never want to add or remove specific features within an image. And if you are going to do something, it is critical that you do it to the entire image. Or if you have a panel of figures, let's say you're doing, you're trying to show fluorescence on a immunohistochemistry panel of cells, you obviously cannot modify one of those images without modifying all of the images. Otherwise, you are manipulating the data, as it were. And I will show some examples of that. But let's say you just want to brighten up the entire image. This is acceptable if you do it across, as I said, the entire image or all the panels of figures at the same time. And you'll see a nice example of that in a little bit. Cropping is okay, but obviously do not crop out any relevant data. You want to show as much of the image as you can. There are some exceptions. You can crop out some things we'll go over later, like unnecessary background, like your fingers are in there, or scales or something like that. And background cleaning can be done. We generally advise not to do it, but once again, it is imperative that you don't modify the actual data within the image if you are going to do something like that. And very, very clear here, if you are going to modify your images, only do this on a copy of your images. Always keep the raw data. Never change your raw data. Always have that separately, because it may be that the journal asks for you to send in that later. Okay, so here is an image of adjusting your white balance contrast. As you can see in the first image, it's a little dark. You might want to lighten it up a little bit. The middle panel shows an acceptable adjustment where you've brightened it a little bit to make it a little bit clearer. The one on the right, they have gone too far and they have adjusted the contrast white balance way too high, and that is not acceptable. You've lost some background data. You do not want to do that. You want to be able to retain the background data. And the bottom this is definitely manipulation, something that's clearly unethical. Here, they actually have modified just the one band at the bottom to make it darker and not the other bands. And this is completely unacceptable. So you definitely want to avoid doing something like that. Okay, next slide. Okay, here's another, which is similar. This is a multi-panel, and what they have done in this from the original image is they have just knocked down the brightness in panel B and left the other ones okay. And this is clearly not ethical, because you're basically showing that there is no staining in that scenario, and that is manipulating of your data. So obviously that's never okay. If you want to increase your brightness, let's say all of these panels were very dark, you can, but you have to increase them all at the exact same amount for all the panels. Okay. And this is obviously not okay ever to do either. This person has just spliced in a cell from another picture and thrown it in there. This is never okay. This is definitely a breach of ethics. Do not add data from other images into your image that you're showing. And then over to Paco.
Thank you. Well, a little bit more of the same. This is an example, and this is well, on the top left there's a reptile there and a whole body, but the other one are histology images. And if you can see the next one, Jessica, see, in this case we just adjusted white balance, and as Elaine said, all the panels. Sometimes in histology, it's okay to work with one of the panels, but provided there's no comparison between panels. Basically, for microscopic images where there's no tissue, you want it to be white, like in this case on the right. On the left is a little bit gray. To me, that's a bad quality image. Next one. This is another example of the same. See that, look at B and look at C, where there's no tissue at the top is gray. We don't like that gray. And the one on the right has been modified to do white balance. In this case, the four panels were treated the same. And next one, please. This is something pretty obvious, but we see frequently. Here we just flip the image. You want to see it. You don't want to have to turn your head to look at the animal. So this is perfectly all right. Next one. This is an example of something that is not acceptable. So in this particular slide, this is we remove a little bit of the glare, so this is okay. But in the next slide, see, you cannot see those red dots. That's hemorrhage. This is the tongue of an animal. This is like Elaine said before, this is absolutely not acceptable, adding data to your picture. We try to reduce foreign material in the photographs. You know, I know you people are from many different disciplines. I'm a pathologist, so we work in the postmortem room, and sometimes you are dirty and you have fingers holding your specimens. So try to reduce that to a minimum, and go to the next example, please. See, this is a photo that is not really acceptable. This is a snake, and the least thing that you see here is a snake. You see that ruler on top, they are in A, you see a hand, you see a caliber on B, and probably the one at the bottom, C, is the only one that shows really something of interest, but try to avoid all that foreign material. Next one, please. This is another one. This is not a bad photograph, but probably half of the image is the gloved fingers. So what I did in this case, the one on the right, is I cropped it so you have the eye, which is what you really want to show, and the minimum amount of gloved finger. And another example, this is a bone from a horse with a bone fracture. And the photo is okay, except that the number at the bottom, that's a case number, and nobody should be interested in the case number. In fact, this might be confidential information that you don't want to publish. So just remove that type of thing.
Okay, I'm going to mention a little bit about Western blots. We have a lot of issues with this. And if you look at a lot of retractions on up here, a lot of them have to do with Western blot manipulations and bad Western blots. So if you are doing these, here are some little tidbits of advice. For one, keep all of your original blots. Absolutely keep them. They don't take up a lot of room. Hold on to them, and hold on to them for a while, because you never know. Try to refrain from doing any kind of adjustments on them except for cropping. And cropping is something that I've seen so many examples where people crop way too close to the bands. I will show some examples, but do not crop too much. You want to see some background on either side of your protein of interest. Once again, I think if you're doing Westerns, you should know loading controls need to be run on every single gel. You cannot use a loading control from one gel and cut it out and put it and use it on another gel when you're showing your figure. It has to be the loading control run on that gel. And always provide a ladder, preferably with two measurements on each, or one on each side of your protein of interest. I've seen too many examples where there was just no ladders. All right, so next slide. So here's just a quick example of something that looks good, the one on the right, minimal cropping. You can see some background. It's not over, the contrast hasn't been adjusted too much so you can't see the background. You can see bands above and below your protein of interest, and there's a ladder. This one looks great. And there's your housekeeping gene below, or housekeeping protein. The one below it has been cropped way too tightly. This is something we don't want to see. We want to see some background on either side. And this one has no housekeeping control either. So please, if you're sending Westerns in, don't send them in these tiny little, they call them, I think, letterbox cropping. Do not do that. Okay, next slide. Oh, okay, I guess we got it. So now we're moving on to legends. Okay, so legends are incredibly important when you're telling the story of your figure. If you don't have a good legend, you're going to lose a lot. We really want to make it that somebody can look at your figure, read the legend, and know what's going on without having to dig through the paper and find that figure and see what's going on. So it's very important that you have a well-written legend. So they must be self-explanatory. As I said, freestanding. So you shouldn't have to dig through the paper, and always check submission guidelines because there will be different font sizes and other things in there. So for each journal, check the submission guidelines. But briefly, at legends, they need to have a brief title, just what's going on. And you want to put a lot of things in there as briefly as possible, but you really want to have like the materials, methods, what controls you use, what statistical tests were used, your p-values, whatever you think is necessary to explain your figure without having to go back to the paper. There are some journals out there who do not want you to put all these things into the actual legend. So make sure you check the guidelines and they will let you know. But generally, you want to have it freestanding, like I said, so they don't have to go back to the paper. You want a brief results statement and all the details in there, like the magnification of the image, the scale, error bars, is a standard deviation or standard error. If there's some strange symbols or patterns in there, make sure you explain it so the reader doesn't have to guess what's going on there. If there's an unusual abbreviation that's not used often, make sure you explain it. So all that needs to be included in the legend. Okay. Next one. And then figure labels. Once again, we want to make these figures as easy for the reader to figure out. So make sure that you add any needed like labeling. ABC should be on the top left. Make sure that's clear. Make sure your arrows are clear. And then the actual placement for what you're looking at, like if it's a cell type or staining, that should be in the bottom left. Not all journals require scale bars. I like to have scale bars for Science Progress and Neuroscience Insights, but if you do put a scale bar, that would be on the bottom right. And of course, make sure you explain the scale bar in the legend. Of course, I think that was it for that. Yeah, I think that was it.
Thank you. Thank you. Such great tips shared. And we've got some great questions rolling in, and we're going to have a great discussion. We really wanted to take the time to focus on your questions because I know we all know this can be a tricky subject of what's acceptable, what's not, and the best practices around this. So before we get into the Q&A, just wanted to remind you of our next webinar, which is focusing on grant fundings, how to secure grant funding. So be sure to join us on June 21st, same time, same place. You'll see us there. And also, there we go. You can see that we have a couple others coming up through the remainder of the year. Go to our landing page for all of the upcoming webinars. We also encourage you to look at our previous webinars, some great sessions in there, including how to select a journal, submission best practices, how to write and structure your manuscript. So even as you're going through right now and working on your images, your figures, your tables, there are best practices that you can follow. And we have other webinars on the structure of a manuscript as well as data and how to present that. We have more coming up in 2024 there, plus a great variety of resources. So with that, we have a great number of questions coming through, and I'm excited to turn it over to the panel for us to take a look at what's coming in. Excited to dive in.
Thank you. Thank you to all our speakers and presenters. This was very useful. And as Jessica said, we have had quite a few good questions here. So I think I'm just going to start. There's been a few questions about if there are any tools that you would recommend for the creation of charts and graphs, if there's anything that is open source and free to use. I'll open it up to everyone here.
I'm trying to think of something that's open source. I've always used PowerPoint and Illustrator, PowerPoint in a pinch when I'm struggling with PowerPoint. But I can't think of, there might be some free versions through Google and things. I'm not sure. Does anyone know?
Would Canva maybe be a good option? You know, that's typically it's not necessarily used for graphs, but it is free for people to sign up, and you could use that as a base to at least load in different images. Sorry, Paco, I think I was about to interrupt you.
No. I was about to say exactly what you just said. Canva is an option. I haven't used it, or I've used it very little. It's a little bit perhaps not as intuitive as PowerPoint. I was going to say Photoshop. Photoshop is not intuitive. But yeah, Canva is an option. There's not a lot of free options out there, unfortunately.
I just want to mention there's a free program called Mind the Graph that will help you create charts and graphs free. Canva has a very peculiar license requirement when you create images with it. So if you're using Canva for creating any visuals, please be careful. Because obviously if you're publishing with a journal where you do not retain the copyright, we don't want to have a copyright battle with Canva at the end of the day. So it's just worth looking into when you're using AI-based apps.
Thanks, everyone. That sounds good. I think there's been quite a few questions here, and this might be, I don't know if we're going to have an answer to this, but I think it's an interesting debate about the use of artificial intelligence in the creation of images and using it in a manuscript. So I don't know if who here would like to start a discussion about this.
I could mention that more with application to text, but certainly Sage has some really good information online on how you credit artificial intelligence, and people are going to be using it for sure. And but you have to, like any other source of information, give accurate attribution. Where did you get this from, and how did it contribute, and who gets credit? So it's something we haven't seen in figures yet that I'm aware of, but certainly in text, people are using that in classrooms now, and writing essays and possibly writing scientific papers. So we're very cautious about that. And we have rules in place. And again, it may vary by journal, but certainly Sage has some good information online from the World Association of Medical Editors, for example, of no, chatbot, ChatGPT cannot be an author, and so on. And you really have to give credit where it is due.
Do we, Adia, I'm imagining, do we want authors to include the prompt they use if they're creating art from a chatbot?
Probably, I think our policy is really catered to text, as Grant has mentioned. I think we would probably say we should avoid using AI-based applications to generate primary data. I think when it comes to visualizations and things, yes, please include the prompts. As far as I'm aware, at the moment, they're not generating visualizations on things like language models because it's mainly text. For example, ChatGPT can't really produce a visualization for you, but if in the future it can, it would be really helpful to see the prompts used so that we can have transparency and a bit more accountability on what's created.
Thanks for that. There was a question here about legends. Someone says, I have been trained not to repeat information from the main text to figure stables. Is this okay to do in the legends in order to make the image understandable?
Yeah, I was struggling writing that slide too, because it's a little confusing putting in the methods. Clearly, you don't want to repeat all of your methods in a legend. You want it to be brief, but and as I said, some journals will tell you to do this, and some will say specifically do not. But when I say putting methods in, you want to make sure that you're putting in enough to explain what the control is, and the cell type would be useful, for instance. You don't need to say like methods of how long, for instance, you performed your, how long you actually, you treated the cells. Actually, how long you treated the cells is actually quite useful. I'm trying to think of something specific that you wouldn't want to put in. I think the important thing here is you want to put in what is necessary to explain your image. So if you are showing something over a time scale of 1 day, 3 days, 10 days, clearly you want that in there. You don't need to put the methods in and how you did it. You just need to make sure you explain the time and the cell types and the different conditions that were used, so you can adequately understand your figure. I think if you're a little unclear on this, like I said, it's a little unsure how much to include. What I would do is look at the journal you are looking to submit to and see what some of the legends look like and get an idea of what people are writing and how much is necessary. That was what I would share.
Thank you. Sorry, that makes sense. Someone is asking about submitting figures. They say figures need to be submitted as separate files. How should the sources or notes for the figures be provided?
I'll say that too. So yeah, we want the images separately, and label them when you put them on figure 1, figure 2, and you will write the legends within your main document. You'll have the legends for the figures at the bottom. So figure 1 and your legend, figure 2 and your legend. So that's usually how people send in their how they submit their forms or they submit their papers. That makes sense. So, but the images should be separate without the legends attached.
And similarly, label the file figure 1, figure 2, but don't put figure 1, figure 2 on the figure itself. That's redundant. So we're trying to keep figures clean and especially graphs. So minimize what is actually within the figure and include it in the legend itself, which, as you say, should be freestanding and understandable.
Thank you. If an image is cropped from the original, should we state somewhere which parts of the image were cropped or why?
No, and unless you remove critical data that you shouldn't. But you show what you want to show, and that's okay.
And just to chime in, that's what I believe it was Elaine that was saying this. You're always working on a copy. So keep that raw image, keep that raw data on file in case that the journal requests to see that later. And then at least you still have that original file and you're using that cropped copied version, and that's what you're submitting to the journal.
And actually, just to speak to that too, if you are doing Western blots, I would go ahead and just submit your Western blots, the original data, alongside as a supplemental material, because it's likely they will ask for it anyways.
Sorry, let me see. There is a question here about language around permissions to use photos. So what is common language for showing you have permission to use photos? Is there a typical way to say that all photos were taken by the author?
I think that's probably a question for you, Jessica.
I can take that. So when every journal submission site is slightly different. At SAGE, there's a question that's asked upon submission saying, do you have permission to use all of the data, the information, the files that you're submitting to the journal in question? If it's your original work, you don't need to indicate that anywhere on your figure. That's implied by you submitting the article as an author. If you're reusing content, that's where you want to indicate the credit, providing that citation, indicating that, you know, this image or this photo is credit of someone, and you'll want to make sure you have obtained those appropriate permissions to reuse that third-party material. That could be actually paying for permission, so going through rights link or the publisher to gain permission to reuse third-party material. If the figure or a table was published under a CC BY license or something similar, that might already be implied that you can reuse it with appropriate credit. There are many different types of licenses and permissions that you might need to obtain. We're actually doing a webinar on that in October. If you want to sign up on different copyrights and permissions and things you'll need to think about there. Sorry, that was very long-winded. The short answer is no. If it's your own original work, you do not need to indicate that specifically in the manuscript. That will be applied with the author byline.
Thank you, Jessica, for that. There was a question here, or a few people have actually asked a question about modifying the DPI of an image before placing it in the article. Is this something that they are allowed to do?
Yes, they are. They are. Once again, in most photographs, modifying the DPI artificially doesn't work. Your photos end up being blurry, and the definition is not good, but you can try, and sometimes they work.
Thank you. I just wanted to highlight as well that we will be sharing the slides after the webinar. So thanks everyone for asking that question, and you will be able to see all the examples that we discussed here more clearly, and you know, at your own pace. So just so you know, you'll be able to look at that later.
I did see a couple of ones in there. Maybe we can write into, they aren't getting the names of the software. So maybe we can write PowerPoint and Photoshop and Canva down so they can refer to that later. And Mind the Graph, is that the one? It's a great name.
It is. Thank you. Someone had asked, should I have to repeat the same information in the legends in many figures if they are the same?
If it's going to be in a separate figure, then yes, you should, because as I said, it should be freestanding. If you can somehow consolidate your figures, so you only have to write the legend once, that might be a better idea. If it's going to be exactly the same information, you might be able to put more panels together.
Thank you. Can you reproduce tables with the necessary references if they are included in paid access journals?
I'll take that one. That just depends on the copyright and the journal. So look at the publisher reuse guidelines if there's any pre-approved permissions and requests. For instance, many publishers will participate in the STM guidelines, and that allows for limited amounts of reuse with citation, with permission. But again, check the publisher specific guidelines around permission, check the copyright of the article, and when in doubt, seek permission. When in doubt, definitely seek permission and provide credit with those necessary references. So that paid for access of a hybrid journal or traditional subscription journal versus open access, you know, it can vary. It just depends on the license and the copyright that is tied to that article. Adia, Elaine, anything I missed or you want to add there?
I would just say again that what you said is, if you are unsure, definitely get the information, the copyright information. It's much safer to err that way than the other way.
Thank you. There was a question here saying some data is taken from different reports and without cropping them, it cannot be used. So what should I do?
I think that's a little unclear. It looks like Paco, you were going to say something. I'm not sure what they meant by data.
I was going to ask the same question, Eileen.
But I think probably what they're asking, and I think Paco already talked about this, if you're cropping out something, if you're keeping the absolute necessary data, then you're okay. You just never want to crop out something that would somehow change the description of your image, or like I mentioned earlier, that your images are data. So you do not want to crop out something that is part of that data. If you're cropping out something that has nothing to do with the data, then you're okay. But treat your images like data.
Yes, if the person who sent the question in would like to follow up on this with a bit more detail, you can reach out to us. You can send your question here, and then we'll answer after the webinar. We'll reach out to you directly, so we can answer exactly what you're looking for. I'm trying to see if there are any other questions here that I have missed. Sorry.
I can jump in.
Yeah.
I see a couple good ones in here, if that's okay.
Go for it.
Yeah. So there was another question around like how to mention a good citation to a figure. And so this goes back when you'll want to follow whatever style guide the journal you're submitting to uses. So some journals follow APA, some follow AMA, some follow Vancouver, Chicago style, and you'll want to use that reference style when providing citations to any articles that you might be referencing. Anyone wanted to add anything on that one?
When all else fails, read the instructions for authors, which usually list. There's a throw in here too from one of the participants. But one of the problems I usually face is even if I submit a very clear image at 300 plus DPI, reviewers complain of image clarity. I suppose it's because reviewers get the PDF version and the TIFF images. So I get within Sage the compiled PDFs, and somehow magically Sage puts together the text and the figures, and they look clear to me. So do you have a comment on that, Jessica, of how the PDFs are compiled and what the expected image clarity is?
Yeah, I would be curious to learn more from this person who submitted the question. What it might be a publisher specific or submission site specific query. At least from the Sage side, we haven't had specific complaints about that. I know from videos, sometimes there's issues with compression, but from images, unless the author supplied image wasn't of high enough quality, I haven't heard of specific complaints like that. Paco or Elaine, is there anything that you might add from a reviewer perspective or image editor perspective?
We get that comment every now and then that the images are 300 DPI. Grant and I see them okay on our computer screens, and then in the PDF they seem to lose resolution. And I honestly don't know what the answer to that question is. I don't know what. It seems to be a rare thing.
Yeah, I see that too.
Yeah, generally I find them to be quite clear. Now, people might not realize, but in the background, of course, as editors, we have access to the original files. But comparing the original files to the compiled PDF, I usually don't see loss of resolution on my screen.
Yeah.
As Paco said.
Yeah, I would say that there's been a few times the PDF has, I mean, it's not as high resolution as the standalone figure general, but it's usually good enough. And if it is showing up poorly in the PDF, it most likely is the original image isn't good enough. So I haven't had anybody complain either, unless I've gone back to the original file and seen that it was also low quality.
And authors should be aware too that the reviewers that we send these manuscripts out to are looking at the PDF. They don't have all of the files that we do, right? They've got the compiled version.
Thank you. Sorry, Jessica, did I interrupt you?
Which maybe leads into another question that was in there about tables. Somebody asked a question about where did the tables go? Well, again, read the instructions, and typically you have references and then tables and then figure legends. And that works really well. Do not embed the tables within the text, even though you'd like them to be right there. Well, we know how to place them. That's what typesetters do. We need the tables at the end, nicely formatted and intelligible, the same as the figure legends. So that's, again, that's instructions for authors are usually clear on that point.
I also, I noticed there were some questions in here around using pictures and images in social science research. And the short answer is yes. Of course, this depends on the research that you're working on and what data that you want to present. And so it may just be a picture or a Western blot, but it might be using line art or creating a graph for a table, which needs to then be converted into an image or figure and ultimately submitted to the journal. And if you're taking qualitative research and converting that into like survey results or interviews or discussions that you're going to end up summarizing in some sort of table or chart, that absolutely can be used in social science research. So today, while a lot of this comes from our own experience in different disciplines, a lot of these same best practices and techniques can be applied to social science research. Following those best practices of, keep those edits to a minimum, be transparent, disclose what you're editing and cropping and what have you. Anyone want to add anything to that? There was another question here that I thought we could address. If an author can create and submit a graph by him or herself, but use another published graph as a template or make a copy of that graph, and even if providing a citation to the original creator. So the graph should be created and generated according to the topic and the research question, rather than using a deductive approach of taking a published graph and change it to suit the current situation. So Adia, thank you for providing some insight on that. So you can, but again, looking at those permissions and credit, that might need to be given based on the situation. Definitely a good question. Adia, please let me know if there's anything you want to add from that from a research integrity perspective.
I guess I would just be very careful of using other people's work as a template or something that you don't currently own as a template. So when you're looking at research data, it should be created by you, something that you can take responsibility for and accountability for. So yeah, just the word template makes me a little nervous, but other than that, it's all good.
Yeah, I would say generate your own graph is probably the best words of advice there.
Mm-hmm.
Thank you. Well, better put than what I said. So thank you for clarifying. Another one I wanted to raise is if you can reuse the figures that you've published, that you as an author have created and published, and maybe in your dissertation or thesis, and reusing that in a future publication. Typically the answer for that is yes, typically. But again, this can vary publisher by publisher. So just check the publisher's author reuse guidelines and the journal reuse guidelines there. This will be very clearly stated on each publisher and each journal's website. And then of course you want to provide credit and cite your original source. So going back to those permissions. Anyone want to add anything there?
I guess just the obvious one, that make sure you're reusing the same thing for the same experimental condition or same topic. So not using archaeology images for citric acid cycles in future research areas. I think that's just that's a very obvious comment, but worth noting.
Speaking of that, I just thought of something too, that when you are putting your figures together, be very, very careful that you're putting the right files and the right images in your figures. There's many times where people have mislabeled their figures unintentionally. It does happen. Be very, very careful when you're putting your figures together that you have the right images.
Thanks everyone for that. I know that we still have a few questions to answer, and we will follow up on those individually with you. So look out for an email from us if you haven't had your question answered here. But I just wanted to thank everyone for joining us today, and to our speakers for this really insightful presentation. We hope you have learned a lot today. And if you have any following questions, you can get in touch with us as well. So I will say thank you to everyone, and we hope to see you next month, and have a lovely rest of your day.
Hey, everybody.
Bye,
Thanks for
everybody.
joining.
Thank you.
Thank
Goodbye.
you.